Hello everyone, this tutorial is for exercise 8, natural selection, Hardy Weinberg, and genetic drift. So from 2 weeks ago, we talked about natural selection. So basic definition of natural selection is a change in the genetic composition of a population across generations driven by differential survival and reproduction. Basically meaning that if individuals in a population have some trait that gives them a selective advantage, they are more likely to survive, reproduce, and pass on that trait that gave them that advantage. Okay, background. One of the things you're going to be talking about a lot in this lab is something called sickle cell anemia. It's a genetic disease. It's a mutation in a gene for hemoglobin. So that is the protein in your red blood cells that carries oxygen. And it leads to deformed sickle-shaped red blood cells, as you can see down here. Those red blood cells can plug up small arteries, they can stick to each other, they cause clots, that sort of thing. They can also cause damage to the spleen, and a number of internal organs. It's also when folks have a sickle cell crisis, it's also extremely painful. So sickle cell anemia is caused by a single gene mutation. Sufferers inherit 2 copies of the sickle cell anemia gene. Individuals with 1 copy of the gene do not suffer from sickle cell, and they are often referred to as carriers. So we need to go over some vocab here. Alleles, those are the possible form of a gene that can occur. So in this case, we're talking about big A being the normal gene and S being the sickle cell gene. Genotype is the sum of the alleles inherited from the parents. Right? Usually you get 1 copy of each gene from each parent. And so in this case, if we have 2 big A's, that person is unaffected. If you get a big A and an S from a parent, from each of your parents, you have the sickle trait, or you're a carrier. And then if you have 2 of the sickle cell genes, 2 S's, that means you have sickle cell disease. The other vocab you need to know is locus, and that's just the physical location of a gene on the genome. Right? So if we say that a gene is located on a certain arm of chromosome 21, that is that gene's locus. Think of it as similar to the word location. So why would this gene persist? Right? It obviously has its disadvantages. You have a 1 in 4 chance of having a child that has serious medical issues and would tend to die at a younger age. The thing is that carriers of this trait have a resistance to malaria. When that parasite, the malarial parasite, which is a protozoa, enters the red blood cell of a person who is a carrier, that cell will sickle and change their shape. Those cells are then filtered out of the body, and so the parasite is taken with it. So those gene carriers have a selective advantage, meaning that they are resistant to malaria. So they are more likely to survive that malaria and go on and reproduce. The downside is that they have a 1 in 4 chance. If they are with someone who also is a carrier, they have a 1 in 4 chance of having a child that has sickle cell disease. Genetic drift. So one of the other ways that the frequency of alleles can change in a population that is different from natural selection is something called genetic drift. And I'm going to make another video to talk more about this. And that's a change in the frequency due to random occurrence. All right, so this very simple example, we've got a 1-to-1 ratio of the blue beetles and the red beetles in this population. A swatter comes down randomly, and now you've got a 10-to-2 ratio after they've reproduced of the red beetles to the blue beetles. Had nothing to do with any sort of selective pressure on either colored beetle. It just happened that way. We'll talk about more examples in another video. So the last thing we're going to talk about is the Hardy Weinberg principle, or equilibrium. This is an idea developed separately by G.H. Hardy and Wilhelm Weinberg. What it states is that allele frequencies are going to remain constant over generations without any outside influence. So if there's no selection pressure or anything like that. The equation is expressed where the frequency of the homozygous dominant phenotype is P2, frequency of the heterozygotes is 2PQ, and the frequency of the homozygous recessive genotype is Q squared. Right? Whole thing is going to add up to 1, or 100%. Hardy-Weinberg equilibrium itself is only going to happen if a population is mating randomly, right? They're not selecting their mates. The population is infinitely large, right? You need a really big population for it. The allele occurs with equal frequency between the sexes. So, for example, in humans, there is potential where there might be a gene that is present on the X chromosome, but is no longer present on the Y chromosome. That means that females would be much more likely to inherit that gene. So that gene would not necessarily be at Hardy-Weinberg equilibrium. Migration, mutation, or selection don't occur. Right? So there's no individuals moving in and out of the population, bringing genes in and out. There is no new versions of the gene showing up through mutation, and there's no selection pressure on particular genes. So let's run through an example. We've got a population of mice. Some are white, some are black, and some are gray. The allele for the black color is going to be, we're going to designate that big B. Right? There's a white color allele, we're going to designate that little b. The genotype of our white mice is little b, little b. The black mice is big B, meaning they've got 2 copies of the gene for black hair color. And our gray mice is big B, little b, meaning they've got 1 copy of the black hair colored gene and 1 copy of the white hair colored gene. So we've got a population of 1,000 mice, and 160 of them are what? Okay, so what can we do with that information? We can use the Hardy-Weinberg equation to predict the frequency of the occurrence of the other alleles and genotypes based on the ones that we know. So we can take a look at this equation. P is going to be the frequency of the big B allele. Q is going to be the frequency of the little b allele. 2PQ is a frequency of heterozygous individuals, ones that have a big B and a little b. P squared is the frequency of the homozygous black coloration individuals, so 2 big B's. And Q squared is the frequency of homozygous white individuals, little b, little b. This is the information that we have. So what we can say is that we've got 160 white mice out of 1,000 total mice. We divide that 160 into 1,000, and that gives us a frequency of 0.16 for our white mice, all right, for that genotype little b, little b. So our Q squared in this case is 0.16. We can then use that to figure out Q by doing the square root, all right, of 0.16. That gives us 0.4. So we now know the frequency of our little b is going to be 0.4. Because the whole equation adds up to 1, we can say that P plus Q is equal to 1. So we can say that P is going to be 1 minus Q, so we minus that from 1, and we get a frequency of 0.6. So now we have the frequency of the homozygous white individuals, all right, 0.16. We have the frequency of that white hair colored gene, all right, or that allele, and that's 0.4, excuse me. And then we have the frequency of the black hair colored gene, which is 0.6. So now we can use that 0.6, and if we square it, because P squared, excuse me, is the frequency of the homozygous individuals that have only the black colored genes, and so that gives us a frequency of 0.36. 2PQ, right? So we plug in the P and the Q there. our 0.6 and our 0.4, that gives us a frequency of 0.48 for our heterozygous individuals. Those are our gray mice. We know our P, we know our Q, all right, the frequency of those two alleles. So now what we can do with this information is our prediction. So we can say, since we know that the frequency of heterozygous individuals or gray mice is 0.48, we can do that times 1,000. So there should be 480 gray mice. We know that P squared, the frequency of those black mice, is 0.36, so we can do that times 1,000, and it says that we should have 360 black mice, based on the fact that we started with 160 white mice. If we then go into this population and we count them up and they are not in those ratios, there's more gray mice or more black mice than we predicted, that tells us that this population might not be at equilibrium, and there may be some sort of selective pressure on this allele or something else going on there. So that is the Hardy Weinberg principle and equation, and an example of how it's used. So let's wrap up. A few things to remember. Humans are subject to natural selection. Some people forget that sometimes. We are still being selected upon. Sickle cell gene carriers are resistant to malaria, and that gives them a selective advantage. Right? And it has to be a pretty good advantage because it comes along with a pretty big downside as well. The Hardy Weinberg equation describes a population at equilibrium where there's no selection and no migration and no mutation. Well, thank you for listening. We'll see you in lab.